Hello and welcome to the third longevity snapshot and first Canadian edition, where we will take a look at a Canadian study which found that reducing a worm's ability to fight free radicals in a specific tissue could lengthen its lifespan. My name is Chris Linnell, the founder and president of the Canadian Longevity Association, Canada's premier longevity nonprofit. If you'd like to find out more about what we do, then please check out the links below this video, where you'll also find a link to the study being reviewed here. Please also give us a like and subscribe to our channel. Now, before we go into the study, a little bit of background on free radicals. So free radicals, also known as reactive oxygen species, or ROS, are produced by our body's metabolism. Now, our bodies have a defense against these free radicals, such as the antioxidants glutathione and superoxide dismutase. The free radical theory of aging, one of the oldest aging theories out there, suggests that the damage caused by free radicals is the primary driver of aging. And if we could reduce levels of these free radicals, then lifespans could be lengthened. There are numerous studies which support the theory. However, there are also a number of studies which contradict it. Indeed, in 2009, the authors of the present study found that knocking out one of those antioxidants, superoxide dismutase, or SOD, could increase the lifespan of worms. There is a link to that study as well as a general overview of what I've just said below this video. Today's study is very much a follow-on to that 2009 paper. Here they're looking at what effects turning on or turning off SOD in specific tissue has on the lifespan of worms. So let's look at the paper. The study comes from the lab of Jeremy Van Ramsdonk from the University of McGill in Montreal. And we'll start by looking at the graphical abstract. The study used C. elegans, a tiny roundworm which only lives for a number of days. You can see a cross-section of the worm here. And by turning off mitochondrial SOD in specific tissue, that led to a rise in mitochondrial ROS, or free radicals. When this was done in the intestine, it led to an increase in lifespan, an increase in heat stress resistance, however, a decrease in oxidative stress resistance. And you can see the survival curve here, with the black line as control, and the red line are the worms which had the SOD knocked out in their intestine. As you can see, the curve has shifted to the right. Now let's take a look at the study in more detail. First, they looked at what would happen if SOD was only turned on in a specific tissue. And for that, they created a series of worms which only had SOD turned on in the various tissues. Now, the worms also had a fluorescent marker when SOD was turned on. And you can see those worms at the top here. So on the left are the ubiquitous SOD worms, which means that SOD was turned on throughout the body. As you can see, it's sort of lit up here. Then there were germline worms. Those are the reproductive parts of the worm. You can see lit up here. The muscles on the outside of the worms, the intestine on the inside, and also the neurons, so the nervous system of the worms. Looking at B here, which shows levels of SOD, on the left-hand side, the WT stands for wild type, so the normal worms, and they were the index or reference values. Now, the strain of worm they used in these studies is called CLK because it showed a greater lifespan effect when SOD was turned off in that 2009 study. And you can see that its levels of SOD were almost as high as the wild type worms. SOD2 worms are worms which had SOD turned off completely, and as you can see, there's no levels of it whatsoever. And in this study, all the worms were a cross between CLK and SOD2, and you can see that those worms also expressed no SOD here at all. Then come the 5 worms we looked at above, and you can see that their levels of SOD were much lower than the wild type. However, they at least had levels of SOD, unlike the CLK1 SOD2 worms. C simply shows how much of the fluorescent marker was expressed in the various worms. And obviously only the worms which were given the marker expressed it. Next, looking at fertility, and the CLK1 SOD2 cross worms show very low levels of fertility when compared to the wild type worms on the left. And the same went for most of the tissue-specific worms, apart from, unsurprisingly, the reproductive or germline worms, which showed higher levels of fertility. However, when it was expressed throughout the worm's body, fertility increased a lot. When looking at the survival of the baby worms, or lethality, as I should say, in wild-type worms, hardly any died. However, the strain used here had much higher lethality, as you can see by this purple bar, which increased even more when SOD was only turned on in the muscle or intestine. However, it was reduced, unsurprisingly, when it was turned on in the germline. You can see by this small red bar here. The post-embryonic development can be seen in F here, with the black line representing the wild-type worms, and all the other strains developed much more slowly. And you can see here the strain used and all the tissue-specific worms are clustered around here on the right. Only the worms ubiquitously expressing SOD developed faster, as can be seen by this blue line here. And looking at movement or body bends, compared to the wild-type worms in white, all the other strains moved a lot less, with the intestine SOD worms moving the least of all. Next, they looked at how the worms would respond to 3 different stressors. First, they were tested against a pathogen, and the worms with no SOD survived much longer than the normal worms on the left. When the SOD was turned on throughout the body, their survival decreased to levels close to the normal worms. In the worms where SOD was only expressed in the specific tissue, their survival increased again, but it didn't reach the levels where there was no SOD at all. The next stressor was heat stress, where they were exposed to 37 degrees centigrade for 3 hours, and the worms lacking SOD survived a lot longer than the wild-type worms. And the same went if the SOD was only expressed in the germline or intestine. In fact, all 4 of the specific tissue worms lived longer than the wild-type worms. Next, they were exposed to oxidative stress in the form of paraquat, which is a very toxic herbicide. And here, the specific tissue SOD worms lived longer than the wild-type worms, with the neuronal SOD worms living the longest. Next, they looked at lifespan, and starting with A on the top left, the wild-type normal worms are the dashed black line. Worms lacking SOD are the gray line. The CLK worms are the black line, and the CLK worms, which also lack SOD, is the purple line. And as you can see, they live considerably longer. Now, comparing the CLK worms lacking SOD, when SOD was turned on back again, it shortened their lifespans in B here. When looking at the specific tissue SOD worms, expressing it only in the germline saw a small increase in lifespan. The same can be said when it was only expressed in the muscle, intestine, and to the greatest extent in the neurons, which is also shown in the bar graph at the bottom here, which is showing mean lifespan or average lifespan of the worms. They then reversed this experiment. Instead of only turning superoxide dismutase, or SOD, in one tissue, they looked at what would happen if they turned it off in only one tissue. And they used RNA interference for this purpose. Now, RNA, among other things, carries the genetic information from our DNA to where proteins are produced to produce those proteins. And by interfering with that process in specific tissue, you can turn SOD off. So in all these survival curves, the black line is the control, and EV stands for empty vectors. So the worms were given the same treatment. However, in the controls, they didn't have that RNA interference. And in A on the top left here, when they gave CLK1 worms this RNA interference throughout their bodies to turn off SOD, they lived longer, as can be seen by the red line being further to the right of the black. When the same treatment was given to wild-type or normal worms, there wasn't any difference, which shows how the CLK1 worms are more sensitive to SOD. On the right in C are worms which have this RNA interference mechanism switched off, so when they were given RNA interference, it made no difference to their lifespan. Moving on to the worms where SOD was turned off in specific tissue, and starting at H here, which is the hypodermis or skin of the worms, and here turning SOD off in the skin seemed to shorten their lifespan, even more so when it was turned off in the muscle here in G. For the neurons and germline and E and F here, there was no statistical difference in their survival. However, when SOD was turned off in the intestine, there was a marked increase in survival. You can see this is comparable to when it was turned off in the worm as a whole, indicating that SOD only needs to be turned off in the intestine to increase the lifespan of these worms. And D here is the same figure we saw in the abstract. Next, they zeroed in on these worms where the SOD was knocked out in the intestine and looked at how they would respond to the same 3 stressors we saw before. So in all 3 of these graphs, wild type is on the left, the strain used in the study, CLK, is here. The strain with the RNA interference ability, second from the right, and where the SOD was knocked out in the intestine on the right-hand side. And when confronted with a pathogen, none of the 4 strains showed any major difference. In fact, it was all not significant. When exposed to heat stress, the CLK and intestine knocked out worm saw a significant and comparable increase in survival. The opposite occurred when the worms were exposed to paraquat, the oxidative stressor. And here there was a comparable decrease in survival between the CLK worms and the intestine SOD knockout worms. Finally, they looked at the function of the worms, starting with the embryonic lethality in A here. And whilst knocking out SOD throughout CLK worms led to a huge amount of lethality, when it was only knocked out in the intestine, it led to a non-significant amount, though still higher than wild-type worms. When looking at fertility, or the brood size, knocking out SOD throughout the CLK worms led to, it looks like no worms surviving whatsoever, whereas knocking out SOD only in the intestines didn't seem to have the same effect. However, again, this was much less than in the wild-type worms. Looking at the development time of the worms, with wild type on the left here, and knocking out SOD in them made no difference whatsoever. When knocked out in CLK worms, it really lengthened their developmental time. However, the difference became non-significant when SOD was only knocked out in the intestine, as you can see down here on the left. Finishing off with movement or thrashing rate, knocking out SOD in the entire worm or only in the intestines made no significant difference. So returning to the graphical abstract, what does this all mean? Well, the study is quite interesting in that turning off superoxide dismutase in a single organ, the intestine, can result in not only an increased lifespan, but also greater heat stress resistance, though it does also lead to reduced oxidative stress resistance, as well as reduced fertility. Now, in terms of the generalizability of these results, you need to remember that this was only done in a single strain of worm, and that further studies are needed to see if this also applies to higher order animals such as mammals or even humans. This is very basic but necessary biology, so who knows where it ends up leading. As for next steps, the researchers here are working on how elevated levels of mitochondrial SOD affect nuclear gene expression, and they are just completing a paper where they've identified a signaling pathway required for SOD longevity that may act to mediate communication between the mitochondria and the nucleus. At the CLA, we wish Jeremy and his crew the best of luck and hope they continue to make major longevity discoveries. I wish there were more work like this in Canada, and the CLA will do everything in our power to encourage it. And that marks the end of another longevity snapshot. Please don't forget to like this video and subscribe to our channel. So until next time, goodbye.